Hello, everyone, and welcome to today's webinar titled Keynote Presentation, Profiling of Receptor Complexes Using BRET. This webinar is part of the 5th Annual Drug Discovery and Development Virtual Conference, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find this live chat located at the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Professor Kevin Pfleger, the Director of Biomedical Innovation at the University of Western Australia and the Head of Molecular Endocrinology and Pharmacology at the Harry Perkins Institute of Medical Research. For a complete biography of all of our speakers, please visit the Biography tab at the left of your screen. Professor Pfleger, welcome. You may now begin your presentation.
Thank you very much, and it's wonderful to be here, and thank you very much for the invitation. So I'd like to take you today on a whistle-stop tour of BRET, which is bioluminescence resonance energy transfer, and how we use that to profile receptor complexes. So BRET is a technology that is become quite widespread now, particularly in the study of G-protein coupled receptors, but also other receptor complexes. And we have our receptor of interest, which is conjugated to a luciferase enzyme from renal luciferase particularly. And we also have an interacting protein typically, which is conjugated to a fluorophore. So that could be Venus, for example. When you have a ligand binding to this receptor in this example, you have the interacting protein come proximal to that receptor. You then have proximity between the luciferase and the fluorophore. As a consequence, you have this resonance energy transfer. And that then results in light emission from the acceptor, which gives us an indication of proximity. We use standard fusion protein cDNA and genetic engineering to remove the stop codon and then have our donor or acceptor constructs on the end of our proteins of interest. We then transfect those into cells, either stably or transiently, and then put them into microplates and read them on a typical instrument. We always measure wavelength windows, so we have the short wavelength emission and the long wavelength emission, and these depend on the different type of BRET that you use. Typically people use BRET1 with a yellow fluorescent protein such as Venus, so they are interested in the long interacting window here, over the short interacting window here, and then they subtract off the background. And that background could be a parallel well of cells which has just the donor present, or typically we use a ligand-induced BRET ratio, in which case we have our control, which is just the vehicle control. So we have our ligand added to one well, we then have this ratio, and then we subtract off the well that has just the vehicle added. Now BRET2, similar principle, but this has greater separation between the two wavelength windows, but with BRET2 we tend to have a much smaller emission, so that results in lower sensitivity of those systems typically, although they can still be used particularly for intramolecular BRET systems. And you can use EGFP, but you just have to be very, very careful about separating those wavelength windows. More recently, the nano-BRET system has come through, and this enables actually transfer of energy across to some very red shifted acceptors. And the advantage of this is that you have much less background here. So if this is the emission from the nanoluke here, then we have the different fluorophores, and you can see that if your fluorophore is actually quite close in terms of the spectrum, you get good energy transfer, but there's more background. And then as you go further away, so these are more redshifted, we get lower energy transfer but less background. So if you go into the redshift where you're peaking at about 630, you have very little background from your emission from the donor. So that's very useful. So what I'd like to do is take you through today various different aspects. So starting with BRET receptor ligand binding assays, then talking about how we monitor receptor signaling and regulation, going through trafficking, particularly looking at cellular localization markers, using the BRET receptor HIT, which is the heteromer identification technology or investigation technology, using that to look at heteromerization, and then looking more at the endogenous protein level using the CRISPR-Cas9 system to BRET tag endogenous proteins. So the BRET receptor ligand binding assays to start with. This is the concept that you actually attach your donor, your nanoluke in this case, to the N-terminus of the receptor, and the BRET event actually happens across to the fluorophore, which is attached to the ligand, and then you compete that off with unlabeled ligand to give competition assay. You can see here that you can do this with green BRET attached here to propranolol, and you can do so also with red. And as I mentioned before, the advantage of red is that you get this much lower background. You can see here this starts at 0, whereas this scale starts at 0.1. And if you actually compare with the scales starting at 0 for both of these, you can see a much higher background with the green, but we still get the same pharmacology. So the take home is you can use a whole range of fluorophores of different colours, but the more redshifted, the less background. There's some illustrations of these assays here, and you can see this saturation here, which has many orders of magnitude. And this is very, very useful aspect of the luminescence technology, because we are using light. We don't have to worry about radioactivity. So if you were doing this with a traditional radioactive assay, this would be a very, very high level of radiation. You just wouldn't be allowed to do it. So we can get many orders of magnitude here because we're using a luminescence-based technology. The other advantage of using BRET is the real-time aspect, and we can actually do these experiments in real time and look at this in terms of kinetic assays and actually see similar KDs regardless of whether we use this kinetic profile or this saturation assay. We can also do this work with peptides as well as small molecules, and you can see this is a TAMRA conjugated angiotensin II ligand, and competing off with a range of antagonists and agonists. So that's binding. So moving on to receptor signaling and regulation. We have arrestin. So a lot of people have done work with BRET using arrestin, and this just illustrates some of the different profiles. And again, illustrates why it's really nice to use real-time assays with the BRET, because this is a wild-type vasopressin receptor, V2R, and you can see with some point mutations, you get some quite striking differences in the kinetic profiles, comparing this one here to quite a different kinetic profile. So we can see a lot of nuance with the BRET system because of the real-time aspects. But of course, we can still do some good old-fashioned pharmacology and look at the concentration response curves. This illustrates the point here. So this is the orexin system, and if you were just to measure initially, both of these receptors would look quite similar. They both come up to the same maximal BRET ratio. However, over time, you can actually differentiate between these two receptors, with the orexin receptor 2 having a much longer and more prolonged interaction between the receptor and the two arrestins. Illustrating the point again, here we have the G protein assay. So this is using an unlabeled receptor, but we've N-luked or nanoluked tagged the G alpha I, and then venous tagged the G gamma 2 subunit. So we're looking at the proximity of these, or in fact the dissociation of these two subunits, and we can see this as a ligand-induced reduction in the BRET signal. Ubiquitin is another molecule that we can look at and see its proximity to the receptor and other molecules. And here you can see, in this instance, we haven't labelled the receptor. Instead, we've labelled the arrestin with the luciferase, and we've labelled ubiquitin with Venus. And you can actually see some quite interesting profiles here, which if you look at this publication, you'll be able to see that we use this to differentiate the two receptors. Okay, so that illustrates the various different ways we can use BRET to look at proximity between the receptor and regulatory and signaling proteins. Now, looking at trafficking. So this was work based on a concept that Nevin Lambert's group came up with, which is that you can actually label a fragment of KRAS that sits in the membrane, plasma membrane of the cell, and you can label various different RAB proteins. And in particular, they pioneered using RAB 4, 5, 11, and some of these down here, 7 and 9. So particularly looking at the internalization process. We also added and developed some markers looking at the forward trafficking as well, which we found very interesting, expanding that to things like RAB1, RAB6, and RAB8, and actually looking at this forward process. So this was all validated in this publication, Molecular Endocrinology, showing how these markers that are labelled with Venus are all in similar compartments to the markers that you would expect to be seen here, so EEA1 and transferrin, lysotracker, etc. So this has all been validated, and it's a nice assay because when you actually add the ligand to the receptor, which is labelled with luciferase, you can actually monitor that trafficking through the different compartments away from the plasma membrane and into the different compartments in real time. So you can see some examples here. This is monitoring the vasopressin 2 receptor trafficking with BRET. You can see it coming down away off the plasma membrane. So this is coming away from the KRAS. So the first panel up there on the top left comes away from the plasma membrane and then traffics into the early endosome and then the fast recycling RAB4 compartment, and then over a longer time scale through the RAB11 slow recycling compartment, and then you've also got trafficking into the lysosome and the late endosome through to the Trans-Golgi network. So you can actually see that the different compartments as the receptor is trafficking through them. And on the other side, you can actually see some of the forward trafficking, which is involving the RAB1 going from the endoplasmic reticulum to Golgi, the RAB8 going from the Golgi up to the plasma membrane, and the RAB6 going through the Golgi apparatus. So, taking you forward in a slightly different slant now, this is looking more at different complexes and heteromerization, and looking at receptor HIT assays, or the Receptor-Heteromer investigation technology. So firstly, let's define what we mean by receptor heteromerization. And there was a group that came together in Bethesda and back in the end of 2008, and then that work was published, although the outcome of those conversations were published in Nature Chemical Biology in this article here. And the group came up with this definition. So the definition is that a receptor heteromer is a macromolecular complex composed of at least 2 functional receptor units with biochemical properties demonstrably different from those of its individual components. So to pull that apart a little bit, it's a large complex. It involves multiple molecules, as we know. These receptors complex with a whole range of different proteins. They could be arrestins, G proteins, various scaffolding molecules. So quite a large complex. There, of course, can be receptors, and there's a lot of debate about what the smallest receptor unit is, but the concept here is a receptor heteromer. You have receptors in there that are functioning in their own right, but when you actually bring 2 receptors together, or more than 2 receptors, those receptors change each other's function. So there's a change in the biochemical properties from this functional interaction. So this isn't just about proximity, this is about a change in pharmacology. So to illustrate here, we've got potentially direct receptor interactions, but we could also have multiple receptor interactions where other proteins are in the membrane separating the receptors that we're investigating. We could also have a situation where we have adapter proteins that are sitting perhaps inside the cell, which are mediating these receptor interactions and actually mediating the allosterism across the complex. So the receptors don't actually have to be touching as long as allosterism can occur across the complex. So why is this important? What are the implications? Well, for a long time we've done a lot of screening in systems where we look at receptors in isolation. We might test a whole panel of receptors, a whole panel of compounds, maybe millions of compounds, and we see a certain effect. But of course, in our cells, in biology, it's more complicated than that. We have complexes of receptors that are potentially interacting and potentially functionally changing the pharmacology. So we could have the effect that we would see in our normal system, our effect A as it were, but we could also be seeing a transactivation of receptor systems. So we could have effect B, which is the normal effect associated with a GPCRB that it's interacting with, or we could have completely novel effects which are specific to the receptor complex. And this is important because we've gone over the last 20 years or so from a situation where we've considered a GPCR as a GQ-coupled receptor, for example, and it's particularly defined by a certain G protein mediated pathway, to this understanding that we have bias and we have the potential for multiple G protein pathways being activated, which arrest in scaffolding particular pathways, and now that the field is now even thinking about how these arrests in scaffolds could actually be resulting in further G protein coupling inside the cell. So the world is always changing and we're always discovering new things, and this is all about the concept of biased signaling. Now, to introduce further complexity is the concept that heteromerization and these complexes can actually result in different coupling of the G proteins, potentially different coupling to arrestins, and a different layer of pharmacology. So this is biased signaling, but it's also through heteromerization. Now, why is that important? Well, this could have ramifications for reducing side effects, because it may be that the reason some drugs have side effects is that we have some unforeseen transactivation of other GPCRs, for example. It has a potential to increase selectivity, because if we understand the pharmacology, it's possible that we could actually use that to increase selectivity. For example, a particular combination of receptors may be expressed at certain levels in a certain tissue, which we're particularly trying to target. The problem, though, is that understanding this pharmacology is technically demanding to assess. So what we've done is actually thought about this and come up with a new approach, and this is called the Receptor-Heteromer investigation technology. And it's quite a simple idea, really, but the idea is that you have a first reporter component on the end of one receptor of interest, but instead of putting the second reporting component on the other GPCR of interest, which is what was traditionally done before, we actually label the interacting group. And then the receptor, when it's activated, the receptor B is activated by a ligand, then the interacting group comes up, interacts with the GPCR complex, and brings the second reporter component into proximity of the first reporter component, and as a consequence there's a signal output. Now, this could be any proximity assay. It could be BRET, it could be FRET, it could be the Dimerix system, enzyme fragment commutation. There's a whole range of different potential assay that could be used for this that measure proximity. And this is the example where we're looking at this from the point of view of an intracellular interaction, when we're looking at this on panel on the left. Panel on the right illustrates how you can actually turn this up the other way and use this to look at ligand binding. So this time we're actually looking at a first reporter component labeled on the N-terminus of the receptor. We then have our interacting group, which is actually a ligand, and that is attached to a fluorophore, which is the second reporter component. The ligand then competes off the interacting group, and therefore you reduce the BRET signal, which is occurring between the first reporter component and the second reporter component on the ligand. So this provides a very nice composition assay, which is heteromer selective. So we've often in the past used luciferase as the interacting group for the first reported component, and then so luciferase is often the first reported component of the system, and then the interacting group in this example is the arrestin, and then the second reported component is a yellow fluorescent protein such as Venus. This illustrates that as you add the ligand that is selective for receptor B or the heteromer, you have this complex here which gives you your signal. However, the other two complexes in this rather simplistic figure are the ones that are sitting in the dark, because you have a receptor A R-Luc homer that is not binding ligand, so is not recruiting your acceptor, and therefore even though luciferase is there, we don't get an energy transfer because there's no acceptor. And over here we have receptor B that is binding ligand, but because there's no donor, there's again no BRET signal, and therefore this complex is also in the dark. So this gives us selectivity for the heteromer complex, and that's the one that gives you the readout. So to illustrate this, here we have the angiotensin type 1 receptor and the CCR2 chemokine receptor, and this is looking at the inhibition of the G-alpha-I activation, because if you actually look at the BRET signal, you see activation of the G-alpha-I by the chemokine receptor in the absence of the angiotensin receptor. So you can see here that you have a concentration response curve as you increase the concentration of chemokine. If you have a maximum dose of chemokine, as you add the angiotensin ligand, nothing happens because there's no angiotensin receptor present. But interestingly, if angiotensin receptor is present, the AT1 receptor, you still get the same concentration response curve as you increase chemokine. And interestingly, if you have a maximal dose of chemokine and then you add increasing concentration of angiotensin ligand, angiotensin 2, you actually inhibit the signal from the chemokine. So we're actually seeing the angiotensin receptor here appearing to actually inhibit the chemokine signal. So that's a really interesting piece of pharmacology. We've also seen this with the arrestin. So here we have the chemokine receptor interacting with the arrestin, and when we have the angiotensin receptor present, when you add angiotensin, you get this small signal here. As you add the chemokine, you get this signal here in blue, but actually when you add both angiotensin 2 and the chemokine, you actually see this more than additive effect. So again, interesting pharmacology that you can see from this assay. And actually what this has done is led us to the idea that you can actually then add two antagonists to block both the chemokine and the angiotensin signal, and it's only when you actually add both that you actually remove the signal down to baseline. And this was the forerunner that led to some preclinical work, and then has now led on to work that's now actually in multiple phase 3 trials, one for kidney disease and 2 for COVID-19. So it's lovely to see this early stage drug discovery now in phase 3 trials, and this is all work done by now being translated by the company Dimerix. So just to expand on this again, this is another combination to illustrate the point. We have the AT1 angiotensin receptor again, but this time looking at the receptor for advanced glycation end products. So this isn't a GPCR, this is a different type of receptor. And here you can actually see we've got our receptor here that's R-Luc8 tagged interacting with arrestin. You've got a nice signal both for the angiotensin receptor recruiting arrestin and the chemokine receptor CCR4. You can see that here. But with the RAGE, that actually only gives you this receptor hit signal with the angiotensin receptor. The CCR4 does not give you the same signal. So this is a nice negative control and showing this selective proximity of and the angiotensin receptor. The trafficking. Again, remembering that trafficking, we're looking at proximity of the receptor to the plasma, trafficking through various different, and then also trafficking back up to the membrane. This is interesting because we tend to see the receptor coming away from the membrane when it's activated, and so we would expect to see the signal with KRAS go down, but in this situation it actually goes up. So that's quite strange, and you would think it's even stranger when you see the RAB5 result, because it does seem to be going into the RAB5 compartment, so it comes away from the membrane and goes into the RAB5 compartment. And then if we look over here, we see RAB11. So this is looking at the panel of the black and red dots in panel E. So we're seeing across to the far right hand side, we're seeing the RAB11 compartment, and that is actually indicating we're going through the recycling endosome compartment. So if you see up on the panel on the left hand side, the receptor is going off the membrane into the early endosome, and then it's going back through the recycling endosome and back up to the membrane. Now, why this is particularly interesting is because, well, firstly, why are we getting an increase in the receptor at the plasma membrane when it seems to be actually going into the cell? Well, the answer to that is looking at this panel E, where you see the RAB8, where that is actually increased quite a lot. And RAB8 is where we're actually getting trafficking from the trans-Golgi network up to the plasma membrane. So if you look at that on the panel on the left-hand side. So in summary, what is happening is, even though the receptor is being internalized and then recycled back through the recycling endosome, you're actually getting an increase in forward trafficking, which means that the net result is an increase of receptor on the plasma membrane. We've also done work in other receptor systems, so not just GPCRs and their friends, but also receptorizing kinases. And this is illustrated here, looking at the proximity to GRAB2. And just to show you some pharmacology here, some concentration response curves, looking at the EGFR proximity to HER3. And this gives them, again, some nice pharmacology. Another illustration here, again a receptorizing kinase, so this is a VEGFR2 on a heteromerization with the beta-2 adrenergic receptor, which is a GPCR, and you can see here that you have the isoprenaline giving you the blue kinetic, and again this illustrates why BRET is very powerful, giving you kinetic data, because you see the kinetic with the iso, and as you then add the VEGF 165A, which is the open circles, so which is the red closed circles, that then gives you quite a different kinetic. So if you contrast the blue kinetic with the red kinetic, you see quite a different profile and a significant difference there. Whereas if you then add the antagonist on top of that for VEGF 165, then you actually have the kinetic that you just saw with the ISO in the first place. So you reverse the change in the kinetic profile that's resulting from the addition of the VEGF agonist. So hopefully that's clear, but it shows you very nicely that you can change the kinetic profile and you can monitor the kinetic profile. And again, if you were just to look at this after, say, about 10 minutes, you'd be forgiven for thinking that the response from these two was quite similar, but you need to see the kinetic profile to see the differentiation, and then it's quite striking. Looking at the heteromerization with the beta-2AR here between the AT1R and the B2R, this is actually looking at the ligand binding assay now, and you can actually see we've got the N-loop on the N-terminus of the beta-2 adrenergic receptor. We're then having a BRET signal energy transfer across to the BODIPI labeled propranolol, and then we compete off with unlabeled propranolol. And you see this saturation curve there on the top right. Now, if you actually have the N-loop on the N-terminus of the angiotensin receptor instead, then you actually have a BRET going across from that NALUC across to the BODIPI labeled propranolol, which is now interacting with the beta-2 adrenergic receptor, which is now heteromerizing with the angiotensin receptor. And again, we compete that off with unlabeled propranolol. Now, the difference here is you still get a saturation assay, but you'll note from the two panels on the right-hand side, the specific binding is about 1/10 of that on the bottom panel compared to the top panel, because remembering that the energy transfer is very dependent on distance, and in fact inversely proportional to the distance to the 6th power. So you actually get a much lower BRET signal because you're getting energy transfer across the complex. Okay, and then in the final 15 minutes or so, I'd just like to take you through the work we've done with CRISPR-Cas9, and this has largely been spearheaded by my postdoctoral fellow, Carl White, who's now actually gone off to work with Dimerix, which is fantastic. And he has really driven and pioneered this use of CRISPR-Cas9 for the BRET system. So here we have the CXCR4, which is endogenous in a HEC293 cell, and this has been labelled with the nanoluke on the C-terminus. So when you add your ligand, you've transfected these cells with CXCR4, and you've transfected these cells with the exogenous beta-arrestin 2 labeled with Venus, but the CXCR4 is endogenous with this nanoluke attached using CRISPR, and it's under endogenous promotion. So what that means is that when you're doing your assays, you're actually seeing some kinetics which are particular to that endogenous protein. This is competed off by the antagonist AMD 3100. Okay, so then we can use this to actually do some quite nice screening. So we have a Z prime here where we have endogenous protein tagged with a BRET donor using CRISPR. And this just illustrates that the nanoluke is much brighter and therefore much more sensitive when you're using very low expression levels. So here we have the Z primes with 0.66 if you've got 25,000 cells per well, or 0.83 if you go up to 50,000 cells per well, and that's using a nanoluke tagged beta-arrestin 2. And this is using exogenous V2R vasopressin receptor labelled with Venus. If you were to actually use the beta-arrestin 2 labelled with R-Luke8 and then use exogenous V2R Venus, you have much lower Z prime because of the much lower light output. As a consequence, it's much harder to detect. We've used this process to look at trafficking. So here we have the CXCR4, which is labelled again with a nanoluke using CRISPR, and then transfecting in Venus labelled KRAS, as well as using the early endosome marker RAB5, for example, and you can see that you get an increase in the proximity to RAB5 as it traffics into the early endosome, and a decrease in proximity to the KRAS. You also get increased proximity to RAB9, RAB11, and RAB7. Now the other thing you can do is actually, let's come through there. The other thing you can do is actually multiplex. So here you have the CXCR4, again nanoluke tagged using CRISPR, but this time we've actually introduced the KRAS with a halo tag, which is red, and the RAB4, which is Venus labelled, which is yellow. So you can actually use an instrument which can actually differentiate between 3 different colours, and actually as a consequence see by multiplexing the decrease in the BRET signal as the receptor moves away from the plasma membrane, so a reduction in the proximity to KRAS halo tag, and also simultaneously an increase in the proximity to RAB4 Venus. So that's very useful to be able to run this in true multiplexing using the same well. So looking at receptor HIT, and this time with endogenous CXCR4 again, using the nanoluke tagged with using CRISPR. Here The exogenous GPCRs are transfected in, as well as exogenous beta-arrestin 2 venous. And there's a couple of illustrations here. One is with CXCR4 and the beta-2 adrenergic receptor. So both of these are classic examples where there's believed to be heteromerization. And you can see that you have the chemokine receptor activated by CXCL12 giving you this signal. You also have the ISO giving you the activation of the adrenergic receptor, again giving you this time a receptor hit signal. But interestingly, when you add both ligands, you get a much more than additive effect. So this is very much illustrating that there is some additional effect as a consequence of heteromerization. This one is more complicated over here on panel B, because you have CXCR4, which is actually activated by both CXCL12 and CXCL11, but CXCR7 is only activated by CXCL11. So that means that if you activate the complex with CXCL11, you just have this quite small green signal, which is the activation of CXCR7 across the complex. You then have, if you activate both receptors, you have this signal here in the blue, which is actually the same signal as the activation with CXCL12, because CXCL12 activates both receptors. So whether you use both ligands or just use CXCL12, you get the same result. So to actually differentiate this particular signal in red, there's no CXCR7 present. So CXCL12 just activating CXCR4 gives you the red signal. So you can see the addition of the red signal and the green signal here is much smaller than the actual additive effect you get. So again, illustrating that you have some kind of heteromer effect here. In the last few minutes, I'd just like to touch on ligand binding using the endogenous promotion system. So here we have in panel B, we have an illustration that if you're transient transfecting in these receptors, so this is an N-loop tagged adenosine A2B receptor tagged on the N-terminus. That, if you put in 5 nanograms, which is actually quite a small amount for transient transfection, you get a very strong luminescence. The kind of luminescence we're talking about with CRISPR, when you've actually got the endogenous receptor, is very, very small. So here you're just looking at less than 6,000 relative light units. So to get that similar level with a transient system, you'd need to go down to 0.05 nanograms, which is a tiny, tiny amount of cDNA. But you can see here, with normalized cytokine accumulation, they all give you similar cytokine results. So HEC293 cells with endogenous A2B, and then these transfected cells, and then the CRISPR A2B. So even though you've got very, very little receptor there, you can still get some really nice saturation assays, and that's illustrated here, and with very nice, clean, flat, non-specific binding. This panel, so that's panel B. Panel C, you've got a ligand there that is not, that is competing off with with the SCH compound that is not selective for the A2B. If you've used a huge amount of it in panel 6, the panel D, sorry, you start to see a little bit of non-specific binding. And panel E is illustrating that if you have a ligand, fluorescent ligand for the angiotensin, then that is obviously not going to bind, and so you get no binding at all. And panel F just illustrates that you can do this with an agonist as well as an antagonist. Now again, with the BRET system, you can do really nice kinetic assays. So you can establish KDs using these kinetic experiments, and also, of course, do some nice concentrated response curves. And to illustrate the comparison with transient transfection, compare this with the next slide, where you can see an experiment that we would normally do with transient transfection with 5 nanograms, you get nice clean data, but if you were to drop that amount to 0.05 nanograms, you get very, very noisy data. So if you compare the CRISPR cells, where you get this nice clean data and some good concentration response curves, similar expression level with transient transfection, you get some quite messy data. And that's because the transient transfection, you're not necessarily having the same concentration of receptor in each cell. So with that, I'd just like to acknowledge all of the people that have done the work. Over many years, we've had great collaborations as well, both academic and industry, and of course, a huge amount of funding to do this work. So with that, I'd like to open the floor for questions. Thank you very much.
Thank you so much, Dr. Pfleger, for that informative presentation. We will now start the live Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's take a look. We already have some great questions coming in. Our first question, when is it best to use nano-BRET compared to BRET1, and when doesn't it really matter?
Okay, so as I showed there, if you're using very, very low expression levels of protein, it's a very good idea to use nano-BRET because it's so much brighter. It's also a good idea to use nano-BRET if you're doing ligand binding assays. When it doesn't really matter necessarily is if you've got good expression levels and you're doing something like an arrestin receptor proximity assay, then perhaps it doesn't matter so much. And also the trafficking assay, again, that's fine with RDK Venus in our hands. But again, you just have to remember if you're going down to very low expression levels, then the nano-BRET is probably your best bet on that one.
Thank you so much. And how does Receptor HIT compare to BRET saturation assays?
Okay, so saturation assays have been used in the field for a number of years, and they give you an idea of the proximity of two proteins in the membrane. So the difference with Receptor HIT is that it tells you about the functional interaction. So because you're adding ligand and you're looking at a recruitment of an interacting protein, it gives you an idea of the functional readout. And it also means that you can use that for screening for heteromer selective compounds as well, which is not quite so easy with the BRET saturation assays, which are quite a lot of work because you've got to get a lot of data points for each of those saturation curves. You can actually add ligand to those and generate multiple saturation curves, and sometimes they shift depending on the presence of ligand or not, which can be useful. But they're a lot more sort of laborious experiments.
Thank you so much, Professor Pfleger, and thank you to our audience members for these questions coming in. Our next question, the audience member says, "Hi, Kevin, great talk. Are you able to look at residence time of the receptors with each other, the ligand with the receptors or the G protein arrestin with the receptors with BRET?"
Yes, absolutely. I mean, there's a lot of work being done in Steve Charlton's doing some nice work with resonance time. So you can generate time constants with the BRET system. I mean, that's really where I think the BRET system lends itself to pharmacology, because it can do this real-time aspect. So absolutely.
Thank you so much. Now, do you have any concerns, Professor Pfleger, about BRET giving signals due to overexpression?
Yes, I mean, I think this is an important point. If you put enough protein into a cell, you're going to get some kind of bystander BRET or artifactual BRET, and that's why the BRET saturation assays were originally developed, to just illustrate that, and why we use Receptor HIT, because that ligand dependency gives you that control. But with all BRET, it's good to have negative controls. It's good to include particular combinations of proteins that don't give you a signal. That gives you some confidence that you're not looking at overexpression artifacts. The other point to make as well is with the trafficking assay, because you're using RABs and they express very well, you're effectively putting a sort of a minefield on that membrane, as it were. So you are utilizing that bystander BRET. So that actually is useful in that respect, but you just have to realize that those interactions or the proximity you're seeing with the BRET is a bystander signal largely.
Thank you so much. And Professor Pfleger, we have one more question coming in from our audience. Which instrument do you use for the BRET?
So we tend to use the Clariostar, and the advantage of that is that because it's got the linear variable filter, you can actually you're not set on sort of specific wavelength windows, and you can actually do a lot of assay development, and that's what we tend to do. So and in particular, you can use that for looking at 3 different wavelength windows, and I illustrated some experiments there where you can actually look at the blue channel for the luciferase, the yellow channel for the venous, and the red channel for the halotag. And it's quite a good instrument for that because you can actually look at all 3 channels. There's also high throughput instruments like the Ferrostar. If you want to do really high throughput, then that can go to that level as well. So so those are the 2 main instruments. Oh, the other one is the Lumistar, which is sort of more just for luminescence, and that can give you simultaneous detection as well. We tend to use Claristar.
Professor Pfleger, thank you so much for your presentation today. Do you have any final comments for our audience before we go?
Well, my main comment would be to say, if you're interested in using BRET, please email me or send me a note through LinkedIn. Very happy to help you out. We have sent constructs all over the world, literally all over the world, and very happy to help you get set up with BRET. I think it's a fantastic technology. I think it's really opened up pharmacology over the last 20 years almost since I've been working in this field. But you know, you've had people like Michel Briviere being trailblazers in this as well, and some great people doing some great work around the world in it. So I think it's a wonderful enabling technology. I think a lot of people are using it. The first review I wrote on BRET back in 2003, I think it was, I managed to find less than 100 articles on BRET on PubMed, and now if you tried to do that, you'd be finding thousands of articles. So that's quite a transition over the last 20 years. So
absolutely. Professor Pfleger, thank you again for your time today and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed for 2 years until February 2024 on that on-demand. Libraries will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.
Thank you.